The adaptive immune system is your third line of defense. This is the part of the immune system that provides immunity to specific substances. It's called adaptive because you adapt to those substances you are exposed to. The adaptive immune system has to encounter the antigen before it can mount an attack against it. This takes time, so your adaptive immune system doesn't start immediately. However, we have plenty of chemical signals that come from the first and second line of defense that sort of warn the third line of defense it needs to get prepared. The adaptive immune system is specific. It is systemic. It's found throughout the entire body, whereas most of your first and second line defense was local. And it has a memory. Once the adaptive immune system has encountered an antigen, it is prepared to attack it every single time it encounters it again. There are two basic types of specific immunity. Humoral immunity uses antibodies. These are produced by the B lymphocytes. Antibodies circulate in the blood. They can bind to viruses or bacteria that are the antigens, inactivating them or marking them for destruction. If you think of this as sort of like a war, your humoral immunity is kind of like chemical warfare. The other kind of specific immunity is cellular or cell-mediated immunity. Here, the lymphocytes have cellular targets like infected cells, cancer cells, or any foreign cells. The T cells are responsible for cellular or cell-mediated immunity. This is more like the hand-to-hand combat unit of the war. Antigens are substances that can mobilize the adaptive defenses. They get their name antigen because they are antibody generating, antigen. Most antigens are large, complex molecules. What's most important is they are unlike any molecule that normally occurs in your body. We say that they are non-self. Complete antigens display immunogenicity. They can stimulate lymphocytes to divide. They also display reactivity. They will then react with the activated lymphocyte. Typically, these are very large, complex molecules. Proteins make the strongest antigens. Haptens are incomplete antigens. They have to be attached to a protein carrier to make them large enough to display all the properties of a complete antigen. A hapten will display reactivity. It will react with an activated lymphocyte, but it is too small to display immunogenicity. That is, it can't stimulate lymphocytes to divide. When it's attached to its protein carrier, then it is like a complete antigen. The molecular antigenicity of any molecule depends on its size and its complexity. Those areas on the molecule that are immunogenic, the ones that react with the lymphocytes, are known as the antigenic determinants. Most natural antigens have a variety of antigenic determinants, not just one, but several. The more antigenic determinants a molecule has, the more potent it is at invoking a response. Different lymphocytes will recognize different antigenic determinants so that one antigen may stimulate several different lymphocyte populations. Large molecules with repeating subunits like hydrocarbons are not antigenic. This is why plastics and Dacron and things like that can be used in the human body without any consequences from the immune system. Now here we see a molecule that is an antigen, and it has 3 different antigenic determinants on it. 3 different lymphocyte populations would respond to this antigen, and 3 different kinds of antibodies would be made to this antigen. We have surface proteins on our cells. These proteins would be antigenic to others. Our immune system is programmed to recognize our self-antigens and not respond to them. The major histocompatibility complex is the group of glycoproteins on the cell surface that are our primary self-antigens. There are several genes that code for these, so that there are millions of combinations possible for the major histocompatibility complex. This means that each MHC is fairly unique to an individual. Each major histocompatibility complex has a deep groove that holds a protein. It can hold either a self-antigen or a foreign antigen. T lymphocytes can only bind to antigens that are presented by these major histocompatibility proteins. The cells of the adaptive immune system are the B lymphocytes, which are the antibody-producing cells. They provide humoral immunity. The T lymphocytes, which do not produce antibodies but provide us with cellular immunity. and the antigen-presenting cells, or APCs. These cells do not respond to antigens, but they are involved in activating the T cells. All lymphocytes originate in the red marrow, but the lymphocytes are educated as they mature. They have to develop immunocompetence. They must recognize only one antigen. It's the antigen they will respond to. Once they have been educated and have immunocompetence, they will display a unique receptor on their cell surface that enables them to recognize their specific antigen. Once the receptor appears, the lymphocyte is now committed to reacting to only one antigen. They're a little bit like Cinderella with a specific shoe size, waiting for their specific Prince Charming. Lymphocytes must show self-tolerance. That is, they have to react to foreign antigens, but not self-antigens. The maturation process is a 2 to 3 day process. It occurs in the bone marrow for the B lymphocytes, and it occurs in the thymus for the T lymphocytes. The selection process is best understood in the T cells. It's a two-step process. First, cells go through positive selection. This is the first test. T cells have to survive self-MHC antigens in order to survive in this test. The T cells must react with MHCs to be activated. Antigen-presenting cells are only going to be able to present their antigen in their MHC groove. Cells that can't recognize self-MHCs are destroyed by apoptosis. The second test is a negative selection test. This ensures that T lymphocytes do not react with MHCs that are displaying self-antigens. Any that do are destroyed again by apoptosis. This ensures that T lymphocytes do not attack normal body tissues. We don't know the B cell education system nearly as well, but we do know that B cells have to learn to be self-tolerant, as well as recognize foreign antigens. Neither process is perfect, so every once in a while there's a little slip up and a T cell gets through that recognizes self-antigens, or we make some cells that don't recognize foreign antigens. The positive selection step means that the T cells have to recognize the major histocompatibility proteins. The major histocompatibility proteins are these outer proteins. So any cell, any T cell that does not recognize the major histocompatibility part, these outer parts, are destroyed. And only the cells that recognize the outer part are saved. Now, the major histocompatibility antigens have this groove in them for holding antigens. At this point, only self-antigens would be in the groove. So in the second step of education, any cell that reacts with a cell protein is destroyed. If it doesn't react with a cell protein, it's kept. So it responds to the major histocompatibility, it recognizes that, but it does not recognize a self protein. That T cell is a keeper. Immunocompetent cells, once they are educated, that's what immunocompetent means, that have not been exposed to their antigen are said to be naive. You can think of them as virgins. Naive T cells and B cells are exported from the primary lymphoid tissue to secondary tissues. You'll remember that the primary lymphoid tissues were the places where the T cells and the B cells became immunocompetent. That would be the thymus and the bone marrow. Once they're immunocompetent, then they can be shipped off to the lymph nodes, the spleen, and those other lymphoid organs. In these areas, they are most likely to encounter their antigen. Lymphocytes make up about 30% or so of the circulating white blood cells. The T cells account for 65 to 85% of those lymphocytes that are circulating in the blood. T cells spend some time daily in the lymph nodes. They'll circulate in the blood, spend a little time in the lymph nodes, and then go back out into the blood. That whole cycle takes about a day for any given T cell. Lymphocyte movement into the tissues is not random but highly specific. There are CAMs, cell adhesion molecules, on the vascular endothelium that help determine where lymphocytes may leave the blood and enter the tissues. In order to be activated, naive cells have to encounter their antigen. This typically occurs in the lymph node, the spleen, or any of the other secondary lymphoid tissue. When the antigen binds to the lymphocyte with the receptor for it, that lymphocyte is selected for further development. This is like Prince Charming with the shoe. There is only one Cinderella that will fit that shoe. So the antigen goes to all of the different lymphocytes trying to find the one that will fit it just right. Once activated, the lymphocyte undergoes rapid cell division to give us an army of cells. Since they all come from the same ancestor, this army of cells is referred to as a clone. Most members of the clone become effector cells, that is, they do whatever that particular population of lymphocytes is supposed to do. But a few members of the clone become memory cells. Memory cells will be able to respond quickly at the next encounter of that same antigen. Our genes determine what antigens we will be able to respond to. Even though we will have hundreds of thousands of cells that can respond to all of these different antigens, only some lymphocytes will ever be activated. Many will never encounter their antigen and will remain naive. Relatively few genes actually control our antibody capability. This is because the few genes that are involved in antibody development are fragmented. As cells make receptors, they put together the gene fragments in different orders. They just keep reshuffling them. They get a new protein every time, a new receptor. This is called somatic recombination. As soon as they have made a receptor, that goes on the cell surface to be expressed as a receptor, and then the gene reshuffles to make a different protein, a different receptor. The antigen-presenting cells are going to be the cells that engulf the antigens and present fragments on their own surface. These fragments of the antigen will be put in that slot on the MHC. T cells can recognize the MHC and will react to the foreign antigen in the slot. Naive T cells can only react to antigens that are presented on MHCs. T cells cannot react to free-floating antigens. The major antigen-presenting cells are the dendritic cells, the macrophages, and the B lymphocytes. Dendritic cells are found roaming under the skin and mucous membranes. They have these long, wispy extensions that kind of sweep through the tissue as they migrate around. Once they encounter an antigen, they ingest it by phagocytosis. Then they get into the lymphatic system to get to the lymph nodes. While they have ingested this by phagocytosis, they have placed a little piece of the antigen in their MHC groove. This means that as they go through the lymphatics, they're presenting this to all of the lymphocytes to see if they can find one that will fit that particular antigen. The macrophages are distributed in the lymphoid tissue and the connective tissue. You'll remember many of them can wander around as they want to. They also engulf antigens and present them to naive T cells. When they present to a naive T cell, not only do they activate the T cell, but they also activate other macrophages. These activated macrophages become insatiable phagocytes. They also trigger powerful inflammatory responses and recruit additional defenses. The B cells are not involved in activating naive T cells. Rather, they have to be activated by already activated T cells. They present their antigen to the helper T cells, and it is the helper T cells that actually complete the activation of the B cell. Activation and differentiation of B cells is the cornerstone of humoral immunity. An immunocompetent but naive B lymphocyte is activated whenever its matching antigen binds to the B cell surface. Now the B cell takes in the receptor and the bound antigen by receptor-mediated endocytosis of this antigen-receptor complex. This is the clonal selection. This means that this cell has now become committed to that particular antigen. Once the B cell is fully activated, something that happens when it associates with the T cell, we will have proliferation and differentiation into effector cells. The effector cells of the B cells are called plasma cells, and they secrete antibodies. When they're going full tilt, they can secrete 2,000 molecules per second. Obviously, a cell working this hard is not going to live very long, so these initial effector cells can only perform for 4 or 5 days before dying. These antibodies circulate in the blood or the lymph, and they will bind to free antigens, marking them for destruction in some form. Any of the effector cells that do not become plasma cells become memory cells. Your primary immune response occurs on the first exposure. There's a little bit of a lag period, about 3 to 6 days, which is how long it takes for the B cells to become activated and to start to divide in large numbers. The antibody levels peak at about 10 days. Because we're going to make some memory cells, the next time we see that antigen, the memory cells are all primed and ready to go. They can respond much more quickly, they give a more prolonged response, and they make antibodies that are even a better fit for the antigen than the original ones. These stimulated memory cells clone to plasma cells, and they make more memory cells. So in our primary response, we have our B cells, and the antigen binds to them. They get differentiated and become activated. They clone into plasma cells and a few memory cells. And the plasma cells make lots and lots of antibody. Then they die. The memory cells hang around. The next time we see that antigen, the memory cells can very quickly clone into a lot of plasma cells that secrete antibody and a few new memory cells. If we look at the antibody response, here is someone was exposed to antigen A here, and here is their first primary immune response with the antibody level. Because of this delay, this primary response, the individual usually shows signs and symptoms of disease. But a little later, if it's exposed a second time, we get a much more rapid response and a much higher antibody level. This is protective. This means that the person will not get sick again. At this point, not only was the person exposed to antigen A, mounting this response to antigen A, but it was also exposed to antigen B, and we see that primary response to antigen B. Here the person would have gotten sick with this particular disease, but been protected from this disease. There are different kinds of humoral immunity, active and passive. In active immunity, the B cells meet the antigen and produce antibodies and memory cells. This can either be naturally acquired, where the organism actually invades the body and disease occurs, but then you have memory cells that mount an effect anytime you see that organism again, or it can be artificially acquired. This is where you receive a vaccine, you mount an immune response to a dead organism or an attenuated organism. Sometimes you have to receive booster shots because the initial response may not give you enough memory cells to be protective the second time you see it. Also, booster shots just revive those memory cells, make sure you have a nice fresh population of memory cells to protect you. With passive immunity, you do not make your own antibodies. So you're not going to have any B cells activated. You're not going to have any memory cells. Here you get antibodies that are already made. Because antibodies are proteins, they're going to break down over time. So this is temporary immunity since the antibodies will disappear. Naturally acquired passive immunity occurs when antibodies cross the placenta from mother to child. They can also be transmitted in breast milk. Artificially acquired passive immunity is when you are given some sort of exogenous antibody made outside of your body but given to you to protect you immediately from disease. For example, snake venom is a toxin. They can give you antivenom that will react against the snake venom that protects you, but if you get snake bitten again, you won't have any antibodies to that snake venom. So this is a temporary kind of immunity. Here you see the humoral immunity. Active, you have made your own. Passive, you got from somebody else. Naturally acquired, you got sick to make your own. Artificially acquired, you were vaccinated to make your own. Passive natural, you got them from your mother. Passive artificial, you were injected with something. Antibodies are also known as immunoglobulins or Igs. Gammaglobulin is one of the major proteins found in the blood. Antibodies are proteins that are secreted by plasma cells, activated B cells. They bind to a specific antigen. There are 5 classes of immunoglobulins, M, A, D, G, and E. MAG is simply a way to organize them and remember them all. Antibody has a basic structure. There's a monomer, a single unit, and sometimes the monomer is the whole antibody. Sometimes there are more than one monomer that make up an antibody. Every antibody monomer has 4 polypeptide chains that are linked by disulfide bonds. There are two long chains that are called heavy chains and two shorter chains that are called light chains. The heavy chains have a hinge region so that you get kind of a Y-shaped to the molecule. One end of the molecule is said to be variable. The other end is the constant region. The variable region is where the antigen will bind. This is variable because each antibody in the class has to be different at that end to bind to the different antigens. But the constant region is the same for every molecule in that class. The constant region is what can bind complement and attach to cell membranes. Here you see the basic monomer, the Y shape, the light chains, the heavy chains, the disulfide bonds. This is the antigen binding site, so this is the variable end. And this part of the molecule is going to be the same for every immunoglobulin in that particular class. This is the part that will bind complement. This is the part that will attach to cell membranes. As we look at the different immunoglobulin classes, IgM is the largest of the molecules. It's a pentamer. It's 5 of those basic Y-shaped units. In the first immune response, this is the molecule that is produced by the plasma cells. This can readily fix and activate complement. IgA is 2 of those units. It's a dimer. This is sometimes referred to as the secretory IgG because it is found attached to your mucous membranes. It's believed that this is the one that may attach to things that try to invade your mucous membranes and stop pathogens from coming into the body. IgD is just the single Y shape, just a monomer. This is found on the surface of B cells. It has something to do with B cell antigen receptors, but we're not totally sure how this one functions. IgG is the most abundant of the antibodies. When you mount a secondary response, this is the antibody that comes into play. So this is the main antibody of secondary and all subsequent responses. It can fix complement, it can activate complement, and this is the largest amount of circulating antibody in your blood. And IgE has as its function that of allergy. This constant end can bind to mast cells. So once we've made IgE and it binds to mast cells, whenever the antigen binds here, it causes the mast cells to release histamine, and that's what initiates an allergic response. Whenever antigens react with their antibody, we get antigen-antibody complexes. These can do different things. Antigen binding to its antibody can cause neutralization. That is, the antibody can bind to sites on viruses or bacterial exotoxins so that they can no longer be effective. Antigen-antibody complexes can cross-link. Remember, most antigens will produce several different antibodies to it. And as all of these antibodies react with the antigen, you can get these large lattice-like things that are produced. That's called agglutination. Sometimes attaching an antibody to its antigen, if the antigen is soluble, makes the antigen insoluble. We call that a precipitation reaction. And then antigen-antibody complexes, you'll remember, fix complement. So that's going to activate the complement cascade, which will initiate opsonization, will increase inflammation, and eventually punch a hole in the cell that started the whole thing. So as we look at what an antigen-antibody complex can do, we've got neutralization, agglutination, and precipitation. In all of these, we have the foreign particle somehow made bigger. This means that phagocytic cells have an easier time finding them and are much more efficient at taking in the particle necessary. So we say all of these enhance phagocytosis. Antigen-antibody complexes also fix and activate complement, and there were the 3 things complement can do. It can act as an opsonin to enhance phagocytosis, it enhances inflammation, and it leads to cell lysis. Cellular immunity is involved with the T lymphocytes. This is the cell-to-cell interaction. This kind of immunity is particularly effective against cells that are infected by viruses or bacteria, cancerous or abnormal body cells, or foreign cells that come into the body, like transplants. Some T cells release chemicals that regulate the immune response. There are several types of T cells, but there are two major populations. They get different glycoproteins on their cell surface. So these cell differentiation glycoproteins cause us to have CD4 cells or CD8 cells. These are the two major T cell types. When activated, CD4 and CD8 cells differentiate into three basic effector cells. CD4 cells become some sort of helper T cell, and there are subsets of helper T cells. Helper T cells can activate B cells, they can activate other T cells like the CD8 cells, and they can activate macrophages. They also are important in directing the adaptive immune response. The CD4 cells are the linchpin of cellular immunity. Without them, not too much happens. The CD8 cells are the ones that become the warrior cells. They become the cytotoxic T cells. These are the cells that go out into the body and destroy any cell that harbors the foreign antigen. Some of the CD4 cells become regulatory T cells. These help moderate the immune response. Of course, there are always some CD4 and some CD8 cells that become memory cells as well as effector cells. The major histocompatibility proteins are involved in activating T cells. T cells can only recognize and respond to antigen fragments that have been processed and displayed on the surface of these APCs. There are two different classes of major histocompatibility proteins. The class 1 MHC proteins are found on all body cells except red blood cells. Remember that each MAC has a groove. In this case, the class 1 groove can hold 8 to 9 amino acid peptides. They're going to hold endogenous proteins, that is, proteins made inside the cell. Typically, proteins made inside a normal cell are normal proteins. So these little endogenous proteins that will be on the MHC groove will be self proteins. The T cells have been trained not to harm those cells. However, if the cell is infected, then foreign proteins may be made as a part of that infection. This is particularly true of viral infections and other parasitic kinds of infections. Also, cancer cells will produce foreign proteins. When this happens, the endogenous protein in the groove will not be self. This will cause the T cell to be activated. Class 2 major histocompatibility proteins are typically only on the surfaces of cells that present to CD4 cells. These are your antigen-presenting cells, the dendritic cell, the macrophages, and the B cells. This groove holds a longer peptide chain, usually 14 to 17 amino acids, and it only holds exogenous antigens, antigens that were made outside the antigen-presenting cell. Since the antigen-presenting cells are all phagocytes, they will have ingested the antigen, processed it, and little digested pieces will be placed in the MHC groove. It is this particular class 2 MHC that binds to the CD4 receptor. So our mature lymphocytes, some of them become CD4s, some become CD8s. The CD8s can respond to class 1s, so they can look at antigen-presenting cells, or they can look at the MHC, not so much on antigen-presenting cells, but on any cell that has a foreign endogenous protein. They'll become cytotoxic T cells and will go out and kill the cell that has that particular protein on it. The CD4 cells are activated by antigen-presenting cells only. And here they are finding their antigen. This is going to be an exogenous antigen. We will make CD4 cells that will get involved. Now, most of these become helper cells. And helper cells are actually involved in activating B cells and CD8 cells. There is an MHC restriction process. CD4 cells can only bind to class 2 MHC proteins, so they're restricted to binding to antigen-presenting cells only. CD8 cells are activated only by antigen fragments on class 1 MHC proteins. So those are on pretty much every cell of the body, and they can respond to any foreign protein there. This is part of the education that occurs in the thymus. Now, sometimes dendritic cells can phagocytize dying virally infected cells. At that point, they can put that foreign protein on the MHC2 receptor, and so we can get CD4 cells that will respond to endogenous proteins, but only if the endogenous proteins have been ingested by an antigen-presenting cell or by one of the macrophages. Cytotoxic cells can then be developed that will attack the infected cell. For T cells to become activated, they too must bind to their antigen. In this case, they have to bind to a major histocompatibility antigen. This is a double recognition system. They have to recognize the self major histocompatibility antigen as well as the non-self antigen. However, this is not enough to get the T cell activated. Binding starts the process, but we must have co-stimulation. One or more co-stimulatory signals are required for this cell to divide. These signals are other molecules that are on the surface of antigen-presenting cells. If binding occurs and there's no co-stimulation, the cell will become tolerant to the antigen and non-responsive. We call this anergy. So here we see a class 2 MHC. It's ingested the bacterial antigen, processed it, and put that bacterial antigen in the groove on the class 2 MHC. The CD4 cell has attached to this. It's recognized the MHC. It's also recognized that this is a foreign antigen. And we have this co-stimulatory molecule that has also triggered the T cell. Now the T cell can divide. It can proliferate into a population of T cells as well as a memory T cell. Once activated, the cell enlarges and proliferates. Cytokines, which we'll talk about in a minute, from the antigen-presenting cells or the T cells help promote this process of proliferation. These cloned cells perform functions according to class. Their primary response peaks within about a week, and then we begin destroying the cells by a process called apoptosis. Starting at about day 7 and continuing until about day 30, these activated cells are destroyed. As we destroy the activated T cells, the effector activity wanes. If we didn't do this, these unneeded T cells could be a potential hazard because in addition to fighting cells, they also make inflammatory cytokines which could continue the inflammation process. The inflammation process is designed to heal. However, if it goes unchecked, it just continues to do cell damage, and this is not good for the body. Cytokines are chemical messengers that are involved in cellular immunity. There are several different cytokines. They can influence cell development, cell differentiation, and various responses in the immune system. A lot of the cytokines are interleukins. Interleukin-1 is released by macrophages, and this stimulates the T cells to liberate interleukin-2. Interleukin-2 is a key growth factor. Without this, we would not have the cells enlarge so they could divide. All activated T cells will secrete one or more other cytokines that help amplify and regulate the many immune responses that go on around them. One of the types of effector T cells are the helper T cells, and the helper T cells play a number of roles. The helper T cell is the central cell in this process. The helper T cell is actually necessary to fully activate B cells. Whenever the B cell encounters its antigen, remember it takes it in by receptor-mediated endocytosis so that it can display that foreign antigen on its class 2 MHC receptors. Once the helper T cell binds to this class 2 receptor, it starts to release cytokines that cause the B cells to divide more rapidly, and also the T helper cells signal the antibody formation to begin. Some B cells can be activated strictly by binding to their antigen. These are called T cell independent antigens, but these responses are usually very weak and short-lived. So most antigens are T cell dependent. That is, we need the T helper cell to complete the activation process of the B cell. And here you see the B cell and the T cell interacting. The B cell has taken in its antigen and is now presented it on its MHC receptor. The T cell binds to the MHC receptor, recognizes the foreign antigen, and then releases all of these cytokines that stimulate the B cell to grow, divide, and produce antibody. This is the complete activation of the B cell. Helper T cells are also important in the activation of the CD8 cells. CD8 cells typically do require a T helper cell to be fully activated. The antigen-presenting cell will present the antigen to the CD8 cell. The T helper cell will also bind to the antigen-presenting cell. This causes the expression of the co-stimulatory proteins that are necessary for the CD8 cell to be fully activated. Once again, an antigen-presenting cell takes in a foreign antigen, and it's going to present it on the class 1 receptor and the class 2 receptor. Antigen-presenting cells have both kinds of receptors. The CD8 cell can bind to the class 1 receptor and start mounting in a response. But it's the T helper cell binding to the class 2 receptor that finishes the job. This is the binding that's necessary to get the cytokines that tell the CD8 cell to proliferate and to go out and find the cell that contains this particular antigen and start destroying it. There are several subsets of T helper cells. The T helper 1 is the one that stimulates inflammation and activates macrophages. This is also the cell that's involved in promoting the differentiation of cytotoxic T cells, the CD8 cells. The T helper 2 is very defensive against parasitic worms. One of the things it does is mobilize eosinophils, and you'll remember that the granules in eosinophils were digestive granules that would help digest away the attachment parts of the parasitic worms. It also activates antibody responses. The T helper 17 is a link between the adaptive and the innate immunity. It releases interleukin 17. Interleukin 17 is what promotes the inflammatory responses. The cytotoxic T cells are the activated CD8 cells. These are your killer cells. They directly attack and kill other cells. Once they've been activated, they roam the body in search of cells that display the antigens that the cell will recognize, the CD8 cell. Their main targets are usually viral infected cells. However, they will also attack tissue cells that are infected with intracellular parasites, cancer cells, and foreign cells. Cytotoxic T cells directly attach to the cell and release chemicals known as perforins and granzymes. These lead to apoptosis of the target cell. Here you see the CD8 cell, the cytotoxic T cell, attaching to the target cell. While it's attached, it releases the perforins, which put little pores or little holes in the cell membrane that allow the granzymes to enter the cell. The perforins and the granzymes are responsible for cell destruction, apoptosis of the cell. Once it's killed that cell, it releases and moves on so it can search for more cells. The regulatory T cells, which are part of the CD4 population, dampen the immune response, either by direct contact or by releasing inhibitory chemicals. This is important to prevent autoimmune reactions. We need to suppress self-reactive lymphocytes outside of lymphoid organs in order to prevent autoimmune reactions. Right now, these regulatory T cells are a current hot research topic because if we can figure out ways to dampen the immune response by using regulatory T cells, we may be able to induce a tolerance to transplants. Now, in looking at the big picture, notice that the CD4 cell is kind of here in the center. This is the linchpin. Whenever an antigen intruder comes in, the innate defenses fend it off as best they can. But if it breaches these, then the adaptive defenses come into play. Some of these antigens can directly activate the B cells. However, this is usually a weak response. But the antibodies will feed back and take care of the intruder. The other thing that happens is the antigen is engulfed by a phagocytic cell, which becomes an antigen-presenting cell, and this will activate the CD4 cells. Now the CD4 cells clone. We get some memory cells, but we also get some helper T cells out of this deal. And depending on what's going on, the helper T cell may go and attach to the B cell, activating it, so we get a much stronger response, more cells, more antibodies to go after the antigen. Or the CD4 helper cells go over and help activate the CD8 cells, and these become the cytotoxic T cells that can go and kill the offending antigen. The CD4 cell is central to all of this. Organ transplants are designed to try to provide a viable organ for someone whose own organ has failed. We have to have similar tissue or else the organ will be rejected. An allograft is the most common type of tissue transplant. This is where we get tissue from a different individual but of the same species. The first thing we have to do is check the ABO and other blood groups and make sure that they're an absolute perfect match. And then the MHCs have to be a close match. Typically, people who get organ transplants are on immunosuppressive therapy. And a lot of this therapy is cytotoxins that will kill rapidly dividing cells. If the immune system has been triggered by this foreign tissue, then some of the most rapidly dividing cells in the body will be those lymphocytes. The idea will be that we kill the population of lymphocytes that would respond to this tissue. Of course, the patient is susceptible to other infections, and so they must be very careful about being around other people. An autograft is when tissue is transplanted from one place to another on the same person. Skin grafts after burns are the most common autograft. Isografts are when you get a transplant from a genetically identical person. To get an isograft, you have to have an identical twin. And xenografts are when we transplant tissue from different species. Now, this sounds a little archaic, but we transplant porcine, pig, heart valves all the time. Immunodeficiencies are either congenital or acquired conditions that impair the production or function of the immune cells or the immune system molecules. One of these is severe combined immunodeficiency, or SCID. This syndrome is congenital. In this syndrome, the individual can't make T cells or B cells, so they have little or no protection against disease. In this particular system, the child has to develop in a germ-free environment and really can never be outside of the germ-free environment. Acquired immunodeficiencies occur when something invades the body and damages the immune system. Hodgkin's lymphoma is a cancer of the B cells. The B cells become unable to act because they are now cancerous. They even become a target of the immune system. Acquired immunodeficiency syndrome, or AIDS, is a viral infection. Here, the virus targets the CD4 cells. And without the CD4 cells, you really cannot mount any new attacks against foreign invading cells. Most people with AIDS die of infections that are viral, fungal, or cancerous in nature. People with AIDS will have their memory B cells, so they will have their antibodies from the past, but they won't be able to make new antibodies or new memory B cells. Autoimmune diseases occur when the immune system loses its ability to distinguish self proteins anymore. This means that the immune system now attacks normal tissue. About 5% of the population has an autoimmune disease, but they're much more common in women. Some examples of autoimmune diseases are things like rheumatoid arthritis. Here the target are the tissues of the joint. This is what the immune system destroys. In myasthenia gravis, the immune system damages the neuromuscular junction so that the muscles and nerves can no longer communicate. This is particularly true of the muscles and nerves of the face and neck. In multiple sclerosis, the immune system attacks the myelin sheath on the nerves. Myelinated nerves are some of the largest nerves in the body. They are responsible for acting very quickly, but they also will fatigue very quickly if they don't have their myelin sheath. People with multiple sclerosis slowly lose the ability to walk, stand, and so forth. In Graves' disease, the autoimmune antibodies attack the thyroid, causing it to overproduce thyroxine. In type 1 diabetes, the beta cells of the pancreas are destroyed. These are the cells that produce insulin. Systemic lupus erythematosus, or SLE, is a disease where collagen in the body is attacked. This can be anything from a relatively mild to a very severe disease, depending upon which tissue the collagen is destroyed in. If it's destroyed in the skin, these people are plagued by unpleasant rashes. If the collagen of the heart is destroyed, they can die very quickly from heart disease. Glomerulonephritis is an autoimmune disease where the kidney filtration membrane is damaged. To treat autoimmune diseases, they try to block the actions of the cytokines by using antibodies against them, or they try to block co-stimulatory molecules. Why would our immune system suddenly turn on us? Well, for some reason, some foreign antigen comes into our body that very closely resembles a self-antigen. Or new self-antigens may appear. This could happen as a result of gene mutation. We could have a change in a self-antigen due to damage, or perhaps because a hapten attaches to one of our normal proteins, and now this hapten is antigenic. Antigens are sometimes hidden during the education phase of the immune system. If some of these hidden antigens become exposed as a result of trauma, that can also trigger an autoimmune disease. In hypersensitivities, the immune system perceives something that's non-threatening as threatening, something like pollen. Immediate hypersensitivities, called acute or type 1 hypersensitivities, are when there is an allergen that stimulates an allergic response. And an allergen is just this perceived threat. It's an antigen that causes an allergic response. Typically, you come in contact with the allergen the first time and no real symptoms occur, but you're sensitized. Your body makes IgE. The IgE coats on the mast cells. The next time you're contacted with this allergen, the IgE binds the allergen. They're already bound to the mast cells, so histamine is released and you get that local inflammatory response. If the allergen for some reason is traveling in your body-wide, you can get a body-wide response. It's called anaphylactic shock. Most of our type 1 hypersensitivities are things like pollen, where you inhale it and only your nasal mucosa reacts. But some people are highly allergic to bee stings. Bee venom would circulate in the bloodstream, causing a massive allergic reaction throughout the entire system. One of the things that occurs with anaphylactic shock is vasodilation, and so you can get vascular shock. So as we look at the sensitization stage here, the antigen, the allergen comes in. A B cell mistakenly responds to it and makes these IgE antibodies which coat up the mast cells. This is sensitization. Nothing happened this time. But the next time you're exposed to that antigen, it's going to attach to the IgE on the mast cells. The mast cells will release histamine, and that's going to give you your dilation of blood vessels, lots of mucus released from mucous membranes, and constriction of the bronchial tubes. Subacute hypersensitivities are caused by IgG or IgM antibodies. It takes them longer to come into play, usually 1 to 3 hours, and they last longer, usually 10 to 15 hours. Cytotoxic or type 2 hypersensitivity is when antibodies bind to antigens on a specific body cell. This stimulates phagocytosis and complement-mediated lysis of the cell in question. An example of a cytotoxic hypersensitivity is a transfusion reaction. Immune complex hypersensitivity or type 3 hypersensitivity occurs when antigens are widely distributed in the body. They're circulating. You get a huge number of antigen-antibody complexes that form. Typically, these should be taken out by the liver, but if they're not taken out fast enough, they cause an intense inflammatory response because these antigen-antibody complexes can trigger complement fixation. Now, lots of body tissues are damaged because of this massive inflammation. The glomerulonephritis of systemic lupus erythematosus is an example of a type 3 hypersensitivity. Delayed hypersensitivities are sometimes referred to as type 4 hypersensitivities. These are mediated by the T cells. These are not antibody mediated. Since T cells are always a little slower to get into the game, it takes 1 to 3 days for these to show up. Here, the T cells bind to the tissue, and you get inflammation and tissue damage. This is because of all the cytokines that are released by the activated macrophages and cytotoxic T cells. Type 4 or delayed hypersensitivities are things like your contact dermatitis. The lymphocytes are produced before birth, and after birth their production continues in the red marrow. The cells become immunocompetent in the thymus and in the bone marrow. A newborn's immunity depends pretty much on antibodies. So the B cells are important and the T helper 2 cells are important because they're the ones that activate the B cells. The T helper cell type 1 gets stronger because the infant interacts with microbes, both harmless and harmful microbes. If you build a good helper 1 system, then you're going to have many fewer problems with allergies in the future. One of the problems is we're currently keeping our kids a little too clean. We give them antibiotics at the first sign of a sniffle or a sneeze. We practically sterilize any food that they eat. It's good for them to eat dirt. It helps their T helper 1 system get stronger. Sometimes factors outside the immune system can influence immune function. The nervous system, for one thing. There is a science called psychoneuroimmunology. This is the link between the brain and the immune system. We know that depression, grief, and emotional stress impair immune responses. Think back to how many times someone has gone through a very long, severe ordeal, a spouse dying of cancer, and they're strong and they're brave and they stay very healthy. But then soon after death, they're found to be very, very sick. Why? Because their immune system was impaired during that severe emotional stress time. Many of you will be fine and healthy through final exams, and the minute finals are over, you will come down with whatever's going around because you've been under the stress of final exams. It turns out that diet may also play a role with the immune system. Vitamin D is required for activation of CD8 cells, so a lack of vitamin D may impair your cytotoxic T cell activity. Also, vitamin D deficiency has been linked to certain autoimmune diseases, like multiple sclerosis. Your efficiency of the immune system wanes with aging. Cancer is much more a disease of aging than anything else, and of course your immune system is part of what protects you from cancer. Also, autoimmune diseases are more prevalent as you age, perhaps an indication that your immune system is a little less efficient at recognizing self versus non-self.